Hello and welcome to this video about genetic crosses. This is designed for the GCSE spec, so if you're watching this looking for the A-level stuff, please refer to the A-level video. So before we begin, let's look at some of the words and terminology we use. So one of the key ones is this idea of an allele. Now an allele is basically a variation of a gene. It's a type of gene, basically. So you've got a gene, for example here gene A, and an allele is the two versions of that gene. Then you've got genomes. Genomes are all of the genetic material inside an organism. And with these alleles, you can have dominant or recessive. And we'll look at why these two are important in a little bit. But these alleles in any individual can be one of two things either. They can be homozygous or heterozygous. So that means if it's homozygous, you have both either dominant or recessive alleles, or if it's heterozygous, you've got one dominant, one recessive. That's what makes something heterozygous. This is, when we talk about this, we are talking about your genotype. So your genotype is the type of gene you have, which is different from your phenotype. Your phenotype is the physical characteristic caused by that gene. So that's the basic question. How do we know what the phenotype will be from the genotype? So in the example here, we have 3 flamingos. 2 of them are pink, 1 is blue. So with the homozygous dominant one, as you can see here, both of them are double A. And if you notice, they've used capital letters. So generally, when we talk about dominant allele, you use capital letters. Whereas your recessive one, you'll notice, are both little a's. So when you talk about a recessive allele, you are giving a lowercase letter. So dominant here is A, the recessive one is little a for coat colour, and you've got heterozygous, which is a combination of both big A and little a. This is where the dominance comes in. So when you talk about dominant, what we're saying is basically if you have one of these, it will show its characteristic no matter what the other one is. So this is just dominance. It's not codominance yet. This is just dominance by itself. So let's say my homozygous dominant flamingo and my homozygous recessive flamingo was to mate and to have a child. So how could you predict what their coat colour would be? So for this, you use something called a punnet square. So a punnet square looks simple. It's like this. It's like a noughts and crosses box, basically. And all you do is you split up the alleles, because obviously when an allele is for a diploid cell, whereas a gamete is haploid, so I'll have one of these. So in the gamete from one parent, I'll have one big A, and for the other one, I'll have little a, little a. So then all you do is you combine them. So big A, little a, big A, little a, big A, little a, big A, little a. So what I would have from this genetic cross, if I had these two parents combining, would be 100% of the offspring would have pink coats. However, they are all 100% heterozygous. So let's now take that a step further. So let's say this time we've got 2 homozygous recessive parents mating. So what colour coat will their offspring have? Well, again, we do the same thing. We set up our punnet square. This time, remember, so this time we're using 2 heterozygous parents. So we use a big A, little a, so we split these gametes up. Big A, little a. And then we'll do the same as we did last time. So we've got 1 big A, 1 big A, 1 big A, 1 little a. So we take from here and here. 1 big A, 1 little a, here and here. And 2 little a's. So this time, if we look at the percentages, we can work out they would have different colour coats. So we'll have 75% of my offspring with pink, 25% blue, and what you have is you've got 1 homozygous dominant, 2 heterozygous individuals, 1 homozygous recessive. So now this brings us on to another idea to do with dominance. So roses are interesting. Roses, you've got white roses, you've got red roses, and you have pink roses. So how does that come from? If we know one's dominant, one's recessive, and the dominant one always shows, how could this be the case? How could you get pink roses as opposed to just white or red? So it's an idea called codominance. So in this time, what you have is, let's say we take a white rose, okay, and we take a red rose. You notice how I haven't used a single letter, but I've superscripted some letters, okay, for the colour. Because if we were to use the same just single letter we used before, it would become too complicated. So this time we use a letter, so for example here C for colour, okay, and then we put a superscript of red or white. And let's say these two parents again crossbred, okay, and we work out the punnet square, so we do the same thing again, okay. So we'll take one of the parents, and what you have, so you've got red, you've got white roses, and we'll do the same thing, CR, CW, CR, CW, CR, CW, CR, CW. This time, because they're codominant, both of these will express themselves. So what we now have is 100% pink roses. So that's the idea behind codominance. It also happens in blood groups, where you've got A, B, blood group AB, blood group O. So that's a little bit more further information. Thank you for watching this video. If you have any questions, please leave them in the comments below.